Hello, this is Zahid Hussain. I'm student of Department of Molecular Biology and Human Genetics. This is the progress report. Uh, this is timeline. I arrived on 4th April and I joined Stem Cell Lab on 12th April. Uh, I arrived late due to visa process and document authentication from Taiwan Embassy, as we in Pakistan do not have Taiwan Embassy. That's why I came late. I started basic lab safety courses and animal center courses. On 1st May, I started cultured to health cell subculture med cell differentiation with flow cytometry and visited to animal center to handle the courses that I've learned are stem cell biology. I learned about stem cells, and this is the subject according to my research interest and labor. Art of tea ceremony and flower arrangement. I enjoyed in this class, and I adopted culture of Taiwan. I learned about socio-humanity and its application in management. This was also an interesting class. Then I attended seminar. I improved my presentation skill. I presented my first seminar. Also, beside these courses, there was a course online, research ethics and education. I completed that course more than 85% marks. So some basic courses that I have learned: animal center course. The aim of this course was we use resistant stress mouse in our future research. That's why I learned about the basic safety and handling procedures and protocols of animal center. I learned how to feed animals, giving injection, taking blood samples, and how sacrifice with anesthesia. I also learned occupational safety training, lab safety training, and their basic protocols with standard SOPs. The techniques that I learned in lab are cell culture, basic knowledge about equipments, reagents, and chemicals. I learned thawing subculture freezing procedures. I learned to make various passages, learned to visualize cell condition under microscope after cell culture, learned to culture cell for differentiation. And I also learned how to handle and how to use flow cytometry. Cell differentiation of health cell. I took health cell as to practice because in future we have to work on the animal cell, because that's why we use a health cell line to culture and to differentiate and to study under flow cytometry. The aims: I use health cell line to practice and observe whether this cell is going to differentiate into megakaryocytes or not. To analyze cell under flow cytometry using cell surface marker CD41B and PI staining, we use TPA. It induced this cell to differentiate into megakaryocyte cells and dissolve, which is dissolved in DMSO. DMSO, when it added alone, it doesn't have effect on cell growth and differentiation. It serves as vehicle control. So far, we can observe that the cell are differentiated into megakaryocytes in TPA group. Here we can see that this is the CD41B marker, and after when we treated cell with TPA and cultured, then we analyzed in flow cytometry. We can have this graph. As we can see here, the cells are differentiated in megakaryocytes, and the amount is 49.45. While two other groups, which are DMSO and control, there is no cell differentiation, and they are absent in TPA. While health control and DMSO group cells have not differentiated megakaryocyte, the graph shows that the cell have differentiated into megakaryocytes. These are the results that I have taken from flow cytometry. So cell differentiated of health cell line, we use propedium iodide to check the polyploidy of megakaryocytes. After manipulating with TPA, the cells were differentiated into megakaryocyte. We use staining propedium iodide to stain the cell and to check the DNA content. The result shows that the few cells have diploid and tetraploid DNA content, while many cells have 4 and polyploidy. TPA treated cells have differentiated into megakaryocytes with polyploidy. So this is how we learn that the cells were the health cell line was differentiated into megakaryocytes, and the DNA content was large. Here in the figure we can see that this is the untreated and this is the DMSO. There no megakaryocytes differentiated. Here we can say no cell was differentiated into megakaryocytes. That's why the half an N4 number can be seen. But here we can see that the TPA, the cells were differentiated in mega karyocytes and the huge amount of polyploidy. We can check the percentage, which is 26.83. This is how the megakaryocytes shows their quality, which is called as polyploid. The conclusions are that cells treated with TP are differentiated into megakaryocytes. Using CD41B cell surface marker, I confirmed that the cell differentiated to megakaryocytes. PI staining confirmed the content of DNA in TPA groups. I learned to handle flow cytometry as it will be used frequently in my future research. So our future work is research project stem cell therapy for resistant stress induced, as we know that acute psychological disorder cause gastrointestinal leakage. Bread gut axis play an important role in the development of gastrointestinal disorder. Current treatment for GI and disorders such as irritable bowel syndrome and inflammatory bowel disease have limitations and safety concerns. That's why we are working on mouse model of resistant stress to study the effect on the gastrointestine. Even as blue dye we used, administration of P-soluble P-selectin is found to rescue stress-induced gut leakage in the mouse model. The administration of soluble P-selectin also leads to an increase in CD34 cells in the circulation. So far, CD34 cells have shown potential in tissue repair after injuries because previous studies evident that CD34 cells can ameliorate liver damage. We hypothesized that CD34 or CD34 P-selectin soluble cells triggered by soluble P-selectin may have therapeutic effects on stress-induced gut injury. This project aims to characterize the rescue effect underlying mechanism of these cells in the stress-induced gut injury. So we are confident and hope that we will characterize the rescue effect and underlying mechanism of these cells in stress-induced gut injury. Thank you.